Reproduction by Mary McLean Healy. What is asexual reproduction? Heredity. The baby baboon below has inherited traits from both its parents. It looks like its parents because of these shared traits. It might grow to be slightly different color or size than its parents, but it will have the same basic form. The passing of traits from parents to offspring is called heredity. Heredity is not just for baboons. All living things, from tigers to bacteria, inherit traits. The environment also affects how living things grow. A tree may inherit the traits needed to grow 12 meters tall, but if the environment does not give the tree the sunlight, water, and soil it needs, it will not reach those heights. Living things can also learn traits. If you inherit traits for athletic ability, you have the potential to be good at basketball, but you still have to practice to learn to play the game. Learned traits are not passed on through heredity. Asexual reproduction. All living things die at some point. For that reason, traits are passed on from parent to offspring to help a species live on. The same traits that help the parent live will also help its offspring. The living thing shown here can reproduce asexually. In asexual reproduction, offspring come from one parent, not two. Asexual reproduction takes place by mitosis, the process by which a cell's nucleus copies and divides. The new offspring have exactly the same DNA as the parent. A bacterium divides into two cells in asexual reproduction. Each one of the two cells is identical to the parent cell. This fast form of reproduction is called fission. Some bacteria can make 2 new cells in 20 minutes. After 40 minutes, there are 4 cells. There are 8 cells in 1 hour. In 8 hours, 1 cell will have multiplied to almost 17 million new bacteria. Most of the time, conditions change, and the bacteria cannot keep reproducing at this rate. Another form of asexual reproduction is budding. Yeasts, some plants, and certain animals can reproduce asexually this way. During budding, a cell in the parent's body makes a small version of the parent. The bud breaks away from the parent to become a new individual. The buds have the same DNA as the parent plant. There are even more kinds of asexual reproduction. Molds reproduce asexually by forming spores. Spores are reproductive cells that are released by an organism. They are often carried off by the wind. They have a hard coat to protect them. When conditions are right, the spore develops. It becomes a new living being. Growing new individuals from body parts is another form of asexual reproduction. Some sea stars reproduce this way. When a sea star loses one of its arms, a new individual will grow from the arm. Some plants use this kind of asexual reproduction too. Buds form on a plant through mitosis. If the bud is cut from the plant, it will grow on its own. Potatoes, such as the one on this page, grow this way. The buds form on the outside of the potato and grow into new plants. Some weeds grow this way as well, growing from a piece of root. How are traits passed on? Structure of DNA. We have to look inside the cell to find out how traits are passed on. Most living things have chromosomes in the cell nucleus. Chromosomes tell the cells what to do, giving directions to different parts of the cell. In multicellular organisms, chromosomes tell cells how to work together to form an individual organism. Chromosomes are made up of proteins and 2 DNA strands that are tightly wound. DNA stands for deoxyribonucleic acid. The cell's nucleus contains the DNA and proteins that make up chromosomes. Usually, chromosomes are spread out in the nucleus. They only come together during cell division. Each E. coli bacterium has one chromosome, which contains 1,000 genes that are made up of 4 billion base pairs. A gene is a series of base pairs or rungs. The number of rungs varies from gene to gene, but at least one known gene has more than 2 million rungs. As you can see in the picture on this page, DNA looks like a ladder that has been twisted. Two scientists, James Watson and Francis Crick, were the first to describe this shape of DNA using X-ray photographs in 1953. Before the 1950s, scientists could see chromosomes in cells, but they did not know the role of DNA. On the ladder of DNA, there are two sides and many rungs. Materials called bases make up the rungs. Each rung is made up of two bases, called a base pair, and the bases are labeled with letters A, T, C, and G. DNA is divided into sections called genes. A gene is a series of base pairs or rungs. Genes control what substances the cell makes and when it makes them, determining the traits of a living thing. The genes in the cells of a rose will contain information about the flower's color. The genes will tell the flower cells what pigment color to make. How many chromosomes? Each species has its own unique number of chromosomes. Members of the same species have the same number of chromosomes. The number of chromosomes each cell of a species has is not related to the size of the individuals. Gorilla, 48 chromosomes, 2 more than each human cell. Crayfish, 200 chromosomes. Horsetail, 216 chromosomes. Copying DNA. 4 bases make up the DNA of all living things. The bases are A, T, C, and G. The order of the pairs changes at different places on the DNA strand. The base pairs are TA, AT, GC, or CG. The way that these pairs are arranged on the DNA strand determines exactly what directions each gene gives to the cells. A series of base pairs, such as TA, CG, GC, TA, give different directions than TA, AT, GC, TA. Different living things have different arrangements of base pairs. Each rung on the DNA ladder is made up of 2 bases. Only certain bases fit together to form a pair. Base A and base T fit together, and base C and base G fit together. Pairing in this way allows DNA to make a copy of itself when it divides. 1. When a cell divides, the base pairs pull apart. One base stays attached to each side of the ladder. 2. Extra bases float around inside the nucleus. 3. The extra bases join the bases still attached to the DNA strands. 4. Because each base only pairs with one other kind of base, the 2 new complete strands of DNA are identical. Take a look at the drawing of DNA below. During mitosis, the base pairs come apart. In the nucleus, there are free-floating bases. These bases pair up with the divided strands. Soon there are 2 full strands of DNA. They are exactly the same as the original strand before it pulled apart. But sometimes an exact copy is not made. One base might take the place of another base. A base might be added or removed. When this kind of change happens, it is called a mutation. A mutation is a change in the DNA strand. In a strand with a mutation, the directions a gene sends can be changed. This gene can be passed on to offspring too. The Human Genome Project. In 1990, a group of scientists set out to map all the genes that make up the human chromosomes. A map of the full set of genes that make up a species is called a genome. By 2003, this team had charted the more than 3 billion base pairs that make up more than 30,000 genes in human DNA. Scientists have been able to identify many genes that are related to hereditary diseases. What is sexual reproduction? Reproduction by two parents. A kitten does not look exactly like its mother. This is because cats reproduce through sexual reproduction, or reproduction by two parents. DNA from both parents are passed on to the kitten. Living things that reproduce sexually have sex cells. These are special cells used during reproduction. In the female, these are called egg cells, and in the male, they are called sperm cells. Each special cell has only half the number of all chromosomes found in regular cells of that species. So if a living thing has 38 chromosomes in all, each sex cell will have 19. Sex cells develop meiosis, when one cell divides into 4 cells. The new cells have half the chromosomes of the parent cell. They are sex cells and can be male or female. A male and female cell join during fertilization. When an egg cell and a sperm cell join, they form a new cell. It is called a zygote. It is the first cell of a new offspring. In time, it will divide into many more cells. Remember that each sex cell has half of all the chromosomes found in the cells of that living thing. When the two sex cells join, the zygote has a full set of chromosomes. For example, when two penguin cells join to form a zygote, the zygote will have half its chromosomes from each parent. Meiosis. 1. Before meiosis, the parent cell makes copies of its chromosomes. 2. The membrane around the nucleus disappears, and twin chromosomes become visible. 3. Pairs of twin chromosomes line up with their copies in the middle of the cell. The pairs move apart. 4. A membrane forms around each group of chromosomes. 5. The cell divides in two. Each has a full set of chromosomes. 6. Twin chromosomes line up in the middle of the cell. They split, and the single chromosomes move to opposite sides of the cell. 7. A membrane forms around each set of chromosomes. 8. The cells divide in two. The cells that result are sex cells. They have only half as many chromosomes as the original cell. Fertilization in seed plants. Flowering plants produce sexually. Their flowers have both male and female parts. The female part of a plant is called the pistil. It often has the shape of a bottle. Flowers can have one or more pistils. The pistil produces the egg. The male part of a flower, called the stamen, produces pollen. Pollen is a powder and contains the male sex cells. During pollination, pollen moves from the stamen to the pistil. The diagram below shows what happens as the pollen moves into the pistil and an egg is fertilized. Flowers have special adaptations that help with pollination. For instance, many flowers have bright petals that attract insects. When the insects move around the flower, they move the pollen too. Flowers that are pollinated by the wind or water have different adaptations that help with fertilization. Fertilization in animals. Like flowers, animals' sperm and egg cells join during sexual reproduction. Depending on the animals, this can take place inside or outside the female. Fertilization that takes place outside the female is called external fertilization. Many mammals that live in or near water use this kind of fertilization. Animals release egg and sperm cells into the water. The sperm cells swim to the eggs and fertilize them. Staghorn coral, shown in the picture, release sperm and eggs into the water at the same time. Then the sperm cells fertilize the egg cells. These animals release billions of sperm and egg cells, but very few fertilized eggs survive because of environmental conditions or predators. For most land animals, fertilization takes place inside the female. Eggs and sperm dry out too quickly on land, and fertilized eggs need moisture. Some land animals, however, do lay eggs. Birds and turtles lay eggs on land. Fertilized eggs need moisture, so these animals' eggs have hard outer shells that keep them from drying out. 1. Pollen lands on the pistil. 2. The pollen grains grow pollen tubes down toward the ovary. Sperm cells travel down to the ovules. 3. Each ovule within the ovary contains an egg cell. Sperm cells reach the egg cells and fertilization occurs. 4. The fertilized egg becomes an embryo. The ovule forms a seed. Individuals differ. In asexual reproduction, each offspring is exactly the same as its parent organism. But in sexual reproduction, every individual is slightly different. Think of a litter of puppies. Each puppy looks a bit different from its brothers and sisters, and each one also looks slightly different from its parents. Every dog has a unique set of DNA because of meiosis. When a sperm or egg cell forms by meiosis, it only gets half a set of DNA. When it combines with another sex cell to form a zygote, it produces a unique DNA combination. Each puppy in a litter will be different because each received a different combination of genes from its parents. For example, one puppy might get its size from the father and its color from its mother, while another might get its mother's size and its father's color. Comparing sexual and asexual reproduction. The simplest form of reproduction is asexual reproduction. It can produce many offspring quickly. Also, only one member from a group of living things is needed for asexual reproduction. Unlike sexual reproduction, asexual reproduction happens with one cell. A cell can reproduce on its own. Asexual reproduction does not require as much energy as sexual reproduction. All living things coming from asexual reproduction have exactly the same DNA as their parents. So a whole group of living things in one place can come from one original parent cell. They will have the same DNA. Suppose there were many amoebas living in a pond. They all have the same DNA, because they all come from a single parent. If there were a sudden change in the pond's conditions, they might not be able to survive. Since all of them have the same traits, all of them would probably die. But with sexual reproduction, each offspring has traits that are a little different from the others. What could kill one might only stress another. That way, when something changes, part of the species will still live on. Those members will pass on the traits that help them live. These traits will help the next group to survive. Asexual reproduction can happen quickly, requires less energy, needs just one parent cell, produces offspring with DNA identical to parent. Sexual reproduction is a slow process, requires more energy, must have two parent cells, produces offspring with unique DNA. How do genes determine traits? Dominant and recessive traits. Genes control the traits that individuals get from both parents. Like the guinea pigs you see here, living things can get one gene for smooth fur from the mother and another for rough fur from the father. Why does a guinea pig with both of these genes have a rough coat? what happened to the gene for a smooth coat. How an offspring looks depends on how the genes from its parents work together. For many traits, an individual has a pair of genes. One gene that masks another is called a dominant gene. The other gene is hidden. It is called a recessive gene. For guinea pigs, rough fur is dominant and smooth fur is not. If a guinea pig has one gene for smooth fur and another for rough fur, it will have a rough coat. In fact, the only way it could have a smooth coat is if it has two smooth coat genes. Sometimes, living things have two dominant and two recessive genes. In the picture you can see that the mother guinea pig has two genes for rough fur. Both are dominant, and they are written as big R and big R. The father has two genes for smooth fur. These are recessive, and are written as little r and little r. When a living thing has two versions of a trait of the same type, as the mother and father below have, they are called purebred for that specific trait. Each offspring will get one trait for a rough coat from the mother, big R, and one trait for a smooth coat from the father, little r, or big R and little r. Because the rough coat is dominant, the young will have rough coats. But what if the parents are not purebred? What if each parent has a big R, little r gene? The young can have three possible gene mixes. They are big R and big R and little r, and little r and little r. Big R and big R, and big R and little r will show as a coat because of the dominant rough coat gene. Only little r and little r will show up as a smooth coat. Sharing dominance. Some gene pairs work differently, such as the feathers on the erminet chicken you see here. This chicken has gene pairs that decide its feather color, but neither gene is dominant, so the chicken has both feather colors. The table on the next page shows how the chicken got its feather color. The table is called a Punnett square. For two parents, it shows all the possible offspring gene mixes. On the side axis is one parent. In the first table, it's a white chicken. The top axis shows the other parent, a black chicken. In the cells, you can see the possible gene combinations. In the first table, two purebred parents, one black, BB, and one white, WW, would produce young chickens with mixed genes. Each possible combination has one gene from the mother and one from the father, BW. So the young will be erminet chickens with both black and white feathers. The second diagram shows what would happen if two erminet chickens mated. Take a look at the possible mixes of genes. What are they? Do you see anything surprising? Gene mixes show up in other ways than they do in the chickens. In the 4 o'clock flowers you see on the next page, the parents have red and white flowers, but the offspring have pink flowers. The genes in this case seem to mix to make a new color. The offspring of the pink flowers can be red, white, or pink. The Punnett square shows how that can happen. This mixing of genes happens to people too. A child may have a mother with curly hair and a father with straight hair. The child could have a blend, wavy hair. But as you saw with the chickens and flowers, two parents with wavy hair could have a child with straight hair, curly hair, or wavy hair. Choosing traits. Corn has been around for a very long time. Take a look at the picture. It might surprise you to find out that the corn we eat is very different from wild corn that grew long ago. In what is now Mexico, about 8,000 to 10,000 years ago, people began to change a grass-like plant they ate. They chose and planted seeds from the plants with traits they liked. Then they grew more plants with the traits they wanted. When those plants grew, they took seeds from plants they liked. They planted those seeds, growing more plants like them. Over time, these plants became a better food for people, and eventually became the corn we have today. Selecting a few living things with desired traits to serve as parents is called selective breeding. And it is not used just for plants. It is also used for reproducing animals. The cow you see is a result of selective breeding. Cows have not always made as much milk as they do today. Some people wanted to get more milk from cows, and others wanted to get milk rich in fat. Cows with these traits have been bred through selective breeding and better nutrition. There are many other uses for selective breeding. It helps make crops that produce more food per plant. Fruits and vegetables are bred to resist disease, pests, and drought. People breed chickens that lay more eggs. People also breed dogs that are smarter, healthier, stronger, and friendlier. Dogs come in many shapes and sizes. You can see two kinds here. History shows that dogs and people have been friends for a very long time. In some ancient cave paintings, dogs are pictured. But dogs have changed a lot since ancient times. Today, there are almost 400 dog breeds. Most of them have been developed through selective breeding, and most breeds were developed after 1850. New breeds were selected because of a dog's special traits, which made them useful for hunting, herding, protection, or looks. Writings and scriptures of the Afghan hound date back to 3500 BC. The Afghans of the deserts of Egypt were bred to hunt gazelle, deer, and leopards. In the mountains of Afghanistan, the Afghan hound was used to guard sheep and cattle. It was also a hunter of small game. The Shetland sheepdog was developed in the Shetland Islands to help herd sheep. The winters there are long, and few plants grow. The farmers of the area developed the Shetland sheepdog to herd sheep and to keep the gardens safe from the flocks.